Does anyone remember what we studied in the previous playlist? Yes? I'm sure no one remembers. So yeah, we covered organelles, we covered mitochondria, endoplasmic reticulum, Golgi, then we did lysosomes, peroxisomes, chloroplast, and the different types of plastids. So one of the organelle which was left and which is like the most important is plasma membrane. So today we are going to study plasma membrane. So what is the plasma membrane? The plasma membrane, also called as the cell membrane, is the membrane that surrounds all the cells. Be it plant cell, be it animal cell, a plasma membrane will always be present. In animal cells, it is the outermost covering of the cell, whereas in plant cells, it lies just beneath the cell wall. So, plasma membrane separates extracellular matrix from the interior of the cell, from the cytoplasm. In this playlist, we are going to deal with what a membrane is, then models of the membrane, composition of the membrane, that is, what is the membrane composed up of. So you all know that the membrane is made up of lipids, proteins, carbohydrates, and some other substances which are found in lesser quantities. So the composition of the plasma membrane, what type of proteins are present in the plasma membrane, what are the type of lipids present, all these we are going to study under this playlist. So here membrane proteins, membrane lipids and carbohydrates. But in this chapter we are only going to deal with basics of the plasma membrane, what is the plasma membrane, the roles of the plasma membrane and the models of the plasma membrane. So these models are important from exam point of view. A lot of questions have been asked from models of the plasma membrane. There are around 6 to 7 models of the plasma membrane which you have to study. So coming to what a plasma membrane is. It's an ultra thin dynamic molecular assembly of proteins, lipids and carbohydrates. So what do you mean by dynamic? When I say dynamic, that means something in the plasma membrane is mobile. It moves. It's not stable. So now we know that the membrane is fluid in nature and that is why we call it as a dynamic assembly of proteins, lipids and carbohydrates. It is a physical barrier between the cytoplasm and the extracellular matrix. As you can see in this small cartoon, there are two sides of the plasma membrane. One is the extracellular side, one is the intracellular side, the cytoplasm. And the molecules which have to enter the cell have to enter through the plasma membrane. And plasma membrane forms a selectively permeable barrier. What do you mean by selectively permeable? That means it allows only selective substances to pass through it. Mind you students, earlier people used to say plasma membrane is semi-permeable, but semi is not the word which is correct. Semi should not be used for plasma membrane. It should be selectively permeable because the plasma membrane selects the substances. It only allows selective substances to pass through it. Now let's look at the roles of the plasma membrane. Some of these roles we have been studying since our lower classes. The roles like it provides protection to the cell, it provides a definite shape to the cell selectively permeable barrier. All these roles we are already aware about. Let's look at these roles in detail. So a plasma membrane has some protective functions. It acts as a container for the cell. It acts as a barrier for the cell. So what this means is that in a plasma membrane, inside it, all the cell organelles, cytoplasm, cytoskeleton, everything is encased. It forms a protective barrier outside the cell. The digestive functions include import of food material and export of waste material. So through the plasma membrane, food material from outside the cell enters into the cell and waste material or the digested material exits the cell through the plasma membrane. Then we have already talked about the selective permeability of the plasma membrane. It helps to transport only certain substances, only selective substances across the plasma membrane. The plasma membrane also has certain receptors on it, and these receptors are responsible for signal transduction. Certain signals come, they land on these receptors, and the message from these signals, these signals can be chemical signals, electrical signals, or anything, and the message from these signals through the receptors are transmitted to the interior of the cell. So plasma membrane has these receptors on it, and therefore plasma membrane plays a very important role in communication or cell-cell communication. It receives signals from outside the cell and relays them into the inner compartments of the cell. Another very important function of the plasma membrane is cell adhesion. How does plasma membrane have a role in cell adhesion? This we are going to study in detail when we study cell-cell communication in Unit 4. But here the only thing you need to know is that there are certain substances present on the plasma membrane, for example cadherins. These substances are responsible for cell adhesion, and these substances are present on the plasma membrane. So this was all about the roles of the plasma membrane. Now we are coming on to a very, very important topic, which is the models of the plasma membrane. Each model is very important. You cannot skip any of the model because questions are asked from models of the plasma membrane. So the very first model is the Overton model. Overton, when he was working with plant root hair in 1902, so he was working on plant root hair somewhere around 1890s, and from 1890s to 1900s he was working on plants and the membranes of these root hairs, and that is when he noticed that there were some hydrophilic molecules which could not enter the cell. So these hydrophilic molecules were repelled by the cell, or they could not enter the cell due to some barrier outside the cell. But when he used lipid soluble molecules, those lipid soluble molecules immediately entered the cell. So why was this so? He concluded that the barrier outside the cell, that is the membranes, are made up of lipids, and they are made up of lipids only. That is why lipid soluble molecules were easily able to enter the cell, whereas hydrophilic molecules were not able to enter the cell. So this was the Overton model of the membrane, which was given in 1902. Coming to the next model, the next model is Irvine-Langmuir model. This model is not very important because questions are very rarely asked from this model, but you just need to know. After Overton model, there was Irvine-Langmuir model. According to this model, the membranes are monolayers. What do you mean by monolayers? So they are a layer of only one, there is only one layer of lipid molecules. We know that membranes have two layers, that is, membranes are bilayers, but according to the Irvine-Langmuir model, the membranes are monolayers. Now, he said that these are two people, Irvine and Langmuir. They said that the membranes are monolayers and they are made up of amphipathic molecules. What do you mean by amphipathic molecules? Amphipathic means a molecule which has a non-polar end and a polar end. So both polar and non-polar sides or polar and non-polar groups will be present in the same molecule. Now we know that lipids, phospholipids are amphipathic molecules. the globular head of the phospholipid is polar in nature, that is, it is hydrophilic in nature, whereas the tails are hydrophobic. So they said that phospholipids or the monolayers have amphipathic molecules. The heads of these molecules interact with water, whereas the tails interact with hydrophobic molecules, because these tails are made up of fatty lipids or fatty acids. The next model is important. It's called the Garter and Grendel model. It was given by Garter and Grendel, two people, in 1920s. They gave this model while working on the membranes of erythrocytes, the membranes of RBCs. So they said that membranes are bilayers. Remember, the first time when someone said that membranes are bilayers are Garter and Grendel. So the Garter and Grendel model says membranes are bilayer, and this model says membranes are bilayer for the first time. None of the scientists before them said membranes are bilayers. But there are certain disadvantages to this model as well. They did not mention anything about the properties of the membrane or the presence of proteins in the membrane. So they said that membranes are bilayers, they are composed of lipids. There are two lipid bilayers, but they did not mention anything about proteins. And remember, students, till this time electron microscope was not discovered. All these studies were carried out without the advent of the electron microscope. The next model is the Davison and Danieli model. This is a very, very important model. After the fluid mosaic model, the fluid mosaic model is the most accepted, most widely known and understood model. After the fluid mosaic model, Davison and Danieli model is the most important. A lot of questions have been asked from this model, so you need to study this model very nicely and you need to understand the disadvantages as well as the advantages of this model. So Davison and Danieli in 1935 gave this model. This model is also called as the sandwich model. We'll study why it is called the sandwich model in a little more detail. So this model invokes the presence of proteins. So this model says that proteins are also present along with lipids in the membrane. But this model also had a lot of disadvantages. It states that membrane is lipid bilayer, coated with a continuous sheet of protein on outer sides, on both the sides. So this is the lipid bilayer, which is coated with a continuous sheet of protein on the upper side or the outer side and a continuous sheet of protein on the inner side. So the proteins are continuous. That means that the lipids are not exposed to the environment. The lipids are completely covered with protein sheets. So they said that each protein layer is 2 to 3 nanometers thick. So this protein layer is 2 to 3 nanometers thick. This one also will be 2 to 3 nanometers thick. And the inner part, the bilayer, will be somewhere around 4 to 5 nanometers. And they said that these proteins are present as beta sheets. So these proteins which completely cover the lipid heads are present as beta sheets. And one thing which they said, which is not correct actually, is that the proteins and lipids are bound together by ionic bonds. We'll see why it is not correct. Now that we have studied the Davison and Danieli model, we have looked at what points did they give about the membrane. Now we come to the disadvantages of this model. So Davison and Danieli, in their model, they said that proteins have beta sheet configurations. That means the proteins, I'll just take my pen. The proteins which are surrounding the lipid bilayer, which are just present outside and inside the on the outer side and the inner side of the lipid bilayer, these proteins are actually beta sheets in nature. Whereas later studies using the electron microscope revealed the presence of alpha helices. So later on, when people studied these membranes under the electron microscope, they noticed that these proteins are actually alpha helices and not beta sheets. So this was one of the disadvantages of the Davison and Danieli model. Now the next thing was that they said that these protein layers, the outer protein layer and the inner protein layer, fits in 2 to 3 nanometers, right? 2 to 3 nanometers of the outer layer, 2 to 3 nanometers of the inner layer, and in between you have lipids which are 4 to 5 nanometers. But the proteins which exist as alpha helices cannot fit well within 2 to 3 nanometers. So it was concluded that these proteins actually are not continuous, are not spread continuously on the lipid bilayer, but these proteins penetrate deep into the bilayers. Are you understanding? So if there are two layers of lipids, outside these lipids the proteins are not continuously spread, but some proteins are outside, some proteins enter into the bilayer. They penetrate deep into the bilayer. Davison and Danieli model further did not say anything about the different kinds of membranes present in different organisms. So the chemical composition of membranes in different organisms varies, right? So the chemical composition of a membrane of erythrocyte will not be the same as the membrane of neurons. Neurons will have more lipids, right? So the difference in the chemical composition of the membranes between different organisms or between different cells of the same organism were not discussed by Davison and Danieli. This was one of the disadvantages. Another disadvantage was that Davison and Danieli said that the proteins and lipids are bound together by ionic bonds. But later studies found out that membrane is dynamic in nature. Membrane proteins as well as membrane lipids are in constant motion. They can move. They are dynamic or fluid in nature. But if there was a presence of ionic bond between the protein and the lipids, the membrane would never have been fluid in nature because ionic bonds hinder mobility. Ionic bonds do not allow the things bound by ionic bonds to move. So the ionic bonds between proteins and lipids will never allow the proteins and lipids to move. Therefore, it was concluded that ionic bonds are not present between proteins and lipids. Rather, these proteins and lipids are bound by hydrophobic interactions, which are much less stronger than these ionic bonds and much less attractive than ionic bonds. Therefore, there is a free movement of proteins and lipids. Next thing was that they inferred that this lipid bilayer is completely coated with proteins. As we just discussed, there are two layers of proteins, one on the outer side of the lipid bilayer, one on the inner side of the lipid bilayer. And this layer of protein is continuous. But when they treated this membrane with certain phospholipases, so phospholipases are enzymes which degrade phospholipids. When they treated these membranes with phospholipases, the membranes were degraded. Now, if the lipids were never exposed to the outside environment, how could phospholipases degrade the membrane? How could phospholipases act on phospholipids? Correct, understood. So, it was concluded that lipids are not always coated completely with proteins. As we just said, these proteins are buried deep inside the membrane at certain points. So, at certain points, the lipids are exposed to the environment, and at these points, the phospholipases could interact with the lipids, the phospholipids, and degrade them. So, this was all about the disadvantages of the Davison and Danieli model. You need to study these disadvantages very nicely because questions are asked from disadvantages as well. They will just not ask you what did Davison and Danieli find out about the membrane or what did they conclude or what their model was all about. They will also ask you which of the following is not a disadvantage of Davison and Danieli model. So questions like these have come. We are going to deal with all these questions in our exam cram section at the end of the playlist. So you need to study these disadvantages also. Now that we have dealt with 4 models of the membrane, that is the Overton model, the Irvine-Langmuir model, the Garter and Grendel model, and the Davison and Danelli model. Now we are going to move on to 2 important models of the membrane which came after the advent of the electron microscope. So these 2 models are the Robertson's unit membrane model. This was basically based on studies carried out on membranes using the electron microscope. So Robertson stained these membranes with a heavy metal. So it's called the heavy metal staining. We are going to do it when we do techniques. So he stained the membranes using this heavy metal osmium and then carried out some studies, after which he deduced a model for the membrane, which is called the Robertson's unit membrane model. After Robertson's unit membrane models, we are going to move upon the most important and the most accepted model of the membrane, which is called the fluid mosaic model. Can someone tell me who gave the fluid mosaic model? Think. It's Singer and Nicholson. So Singer and Nicholson gave the fluid mosaic model and we study it till date. It's not the latest membrane model. There has been another model by Irvine and Henderson. That also we are going to deal with in the next lecture. So coming to the unit membrane model, it was given by Robertson in 1950s. It is actually an extension of the Davison and Danelli model. So Robertson, he was working on membranes from different organisms. He wanted to study the composition, the structure of these membranes, and he carried out heavy metal staining of these membranes. Just a second, let me use my pen. Yes. So he carried out heavy metal staining using osmium tetraoxide. Osmium is a heavy metal. So he stained these membranes from different organisms and observed that these membranes had a trilaminar structure. So there were 3 layers in the membrane, and there were 2 dark osmiophilic bands. One was the outer band, one was the inner band, and these 2 dark bands were separated by one light osmiophilic band which was present in the center. So the 2 dark osmiophilic bands represented proteins, whereas the one light osmiophilic band in the center represented membrane lipids. This structure, the entire structure which he visualized upon heavy metal staining, looked like a railroad track structure. So can you see? Similar to a rail track. These are the outer sides of the rail track and these are the wooden planks in between the rail track. So it looks like a railroad track structure. So Robertson called it as the railroad track structure. It is also called as the trilaminar structure. So after Robertson's model, we come to the most important and the most recent model of the membrane, the Singer and Nicholson model, which was given by Singer and Nicholson in 1972. It's also called the fluid mosaic model or the icebergs in the sea model. Actually, fluid mosaic model is not the latest model of the membrane. One more model has been given after the fluid mosaic model, which is called the Irvine and Henderson model. But till date, fluid mosaic model is the most accepted model of the membrane. So let's see what this model states. So Singer and Nicholson proposed that the membrane is a mosaic of proteins dispersed within the bilayer. So proteins are like mosaics. They are like icebergs dispersed in the sea of these of the two layers, the bilayer. So the bilayer is mobile. In between the bilayer, in between this sea, proteins are present as mosaics. The hydrophilic regions of these proteins So these regions are exposed to water. It is exposed to the outside environment, whereas the hydrophilic regions of these proteins are buried inside the membrane because they will interact with the lipid tails. The lipid tails are hydrophobic. Therefore, only the hydrophobic parts of the proteins can be buried inside the plasma membrane, inside the bilayer, sorry. So, fluid mosaic model states that phospholipids are the most abundant lipids in the plasma membrane. These phospholipids have two parts. They are amphipathic molecules. They have two parts. One is hydrophilic and one is hydrophobic. So the non-polar regions of the lipid molecules face the core and the polar head groups face outward. That is, the polar head groups interact with extracellular matrix or the hydrophilic environment outside. And the proteins are embedded inside the bilayer and they are held by hydrophobic interactions. This was all about the fluid mosaic model. Next lecture we are going to study fluid mosaic model in some more detail and we are going to study the membrane composition. So this was just the introduction of the fluid mosaic model. In the next lecture I am going to deal with the fluid mosaic model in detail, the different components of the plasma membrane. So there are integral membrane proteins, there are peripheral membrane proteins, the types of lipids, carbohydrates, cholesterol. I'm going to deal with everything in the next lecture. So do not forget to subscribe to Utkal Digital Classes. Stay tuned for the next lecture. Please press the bell icon so that you get all the notifications and happy learning.